Thank you very much, Chairman. Welcome back, everybody. I thank you very much, the organizers, and particularly Dr. Patricia Rillo for her kind invitation to this meeting, which is so far very diverse and very interesting talks. And I have to thank Patricia also for giving me the best spot to talk. You are fully awakened now. You had your coffee. You don't have any excuse to pay full attention. So the problem when we work with natural products is that we deal with very complex and chemical entities. We have hundreds of compounds in there, and we don't know which one is the bioactive, and many times when you start doing a bio-guided isolation of the bioactive compounds, you end up with nothing, or with very low activity compounds. Nevertheless, the complex chemical entity, which is the natural product, many times have powerful activities, and this is what we want. But we need ways to standardize or predict or measure this activity. This is what I will try to explain in this small talk, how we can apply modern tools like artificial intelligence or advanced statistics, which are nowadays very easy. You have a computer, you have a software, and you can use it without knowing how to program a computer, like 20 or 30 years before, to predict the bioactivity of a natural product without even going to the lab and doing the experiment, or to find the bioactives or the likely candidates for the bioactivity within them without going to the lab and doing the bioactive isolation, or even interpret the use that other cultures make of natural products and translate them into our molecular knowledge. How we can predict? Well, there's a potent tool which is called artificial neural networks, which is a computer algorithm that completely mimics the function of a neuron. You know, a neuron receives a stimuli, and then they process the signal, and if the signal is strong enough or meaningful enough, the neuron will, it's a bit temperamental, the neuron will decide to propagate this and to other neuron to propagate this signal. We have in the computer, we can create an algorithm where the inputs that may be the composition of your natural product can be computed, and if the sum of the inputs is higher than certain threshold, then you have a function which converts this threshold into an output onto another neuron. You know that neurons doesn't work alone in isolation. We need neural networks. So we need the stimuli to stimulate several neurons that are outside what we call the input layer. Then there's many neurons in between that process this signal and convert it onto a output, onto a response of the neuronal system. The same we can do with the computer. We have all these algorithms distributed in several layers. The first one receives the input, as I said, probably the composition of your natural product, and the last one will predict the bioactivity you would like. How they do this? Well, for a human it's very easy. Imagine that you have these three numbers and you want to produce 3. For us it's very easy. You can do 4 minus 1, or 2 plus 1, or 4 minus 2 plus 1. For us it's very easy. We have millions of neurons and work very, very fast. For the computer is a bit of a trial and error. It has to first try randomly some computations and then get a first result, and then it will compare 1 with 3 and go backwards and adjust the algorithm until you get closer and closer to the value you want, and then you stop the training And then you get a neural network that predicts what you want. So how can we use it, for example, to predict the antioxidant and the antimicrobial activity of essential oils? You know, essential oils have been around in humankind for thousands of years. They have a lot of antioxidant activity and most important antimicrobial activity. But because each essential oil may have 100 components, it's very difficult to make a use, or even they vary the composition from one year to another year, and we don't know if the bioactivity will be the same or not. We try to browse the literature for all the data that the scientific community is producing about essential oils and antioxidant activity, for example. There are hundreds and hundreds of papers. So it should be possible to get a composition of all these essential oils, to get the results on the antioxidant assay, and train the computer, and then we don't have to go to the lab anymore. You just train the computer, and the next essential oil you isolate, you analyze it, input the composition, you get the bioactivity. The problem is that the scientific community is working in disarray. We don't work properly. We don't work as a community. Each one is using a different reference compound, a different method, a different variation of this different variation of the method. So actually, of the hundreds and hundreds of papers, we could really retrieve very little information that was comparable. Because if you input noise to the neural network, you get noise. You input rubbish, you get rubbish. So, for example, we were, after 2 months of browsing literature, we could end with 32 essential oils that match the same protocol, the same reference compounds. We were sure they were comparable. So what we do is take 27 of them to train the computer and 5 to prove that the computer is working, actually. The problem is essential oils, all these essential oils had up to 80 compounds, and neural networks have a limitation. You cannot input 80 compounds, it's too much still for them. So we had to go down to 30 compounds, and this is the hard work. We have to figure out which compounds are minor, so we don't use them, which compounds are known to have an activity, so we keep them, and select a good variety of the compounds that are in the middle, that are maybe not known to work, but too important in the composition to be obvious. So we've got this set, and actually for each of these neurons, you have the percentage in the essential oil of this molecule here, and then it will compute, it will compute, and it will create a model to predict the antioxidant capacity of this oil. So once you have this trained, the next researcher that produces an essential oil and analyzes it only has to input it in our neural network, and we'll get the value in the PPH assay or in the linoleic acid assay. There's 2 different assays. One is free radical scavenging, another one is lipoperoxidation. And you get the prediction of the IC50 with an error of 1% to 3%. You don't bother to go to the lab again anymore. That's the power of the computational. Many of you will say, well, that's easy. Antioxidant activity is very well related to phenols, the antioxidant phenolic compounds. Actually, it's not. In essential oil, you see the linear training between phenol content and antioxidant activity in all these essential oils is not significant. There are a lot of antagonisms, synergisms, and additivity in these essential oils. That was easy to model a chemical reaction, because eventually it's a chemical reaction between many chemicals. Could we model the response of a living organism to many compounds? Well, we tried to do it. We chose a gram-positive, a gram-negative, and a bacillus also gram-positive anaerobic, and Candida albicans as a representation of a fungi. And we did the same. We tried to look for literature and comparable results. That's really the difficulty we have. Each laboratory is using a different antibiotic of reference. Each one is doing a different modification of the same protocol, and it's very difficult really to. So we end up with 22. We have also to reduce 180 compounds from all these essential oils into a set that the neural network could digest. So we end up with 22 principal antimicrobials with a limit. They had to have at least 5% within the oil. We have several restrictions to make it to the set. And then you see, for example, for Staphylococcus aureus, you have in blue the prediction of the machine and in pink the real value. And then you see that some are better than others. You do 3 independent experiments with three different validating sets. And finally you get a model which more or less within the errors of the experimental model predicts the antimicrobial bioactivity of these 22 compounds at the same time. It's very funny because actually the neural network reflects the complexity of the microbes. It was easy, let's say, to predict Staphylococcus aureus and Clostridium, but it was less easy to predict Escherichia coli and Candida, and you know that Escherichia coli and Candida are very difficult to treat, actually. We have more resistance, and Candida albicans is a fungi, it's very difficult to tackle it. Can the neural network predict 2 activities at the same time? Yes. And even got better, we don't know, maybe because the set of data was much better in this case. But the neural network can predict, for example, Staphylococcus aureus and Escherichia coli at the same time for the same set of compounds. And 3, yes, also. So what we have here is that artificial neural networks is probably a tool we need to explore more in order to start stopping, going to the lab and doing the same and the same again whenever we have already a lot of data to predict the bioactivity. But the neural network is not a crystal ball perfect. Will give you the value within the error of the model, and our models have quite a big error sometimes. That's very good. And how could we, instead predicting the bioactivity, finding the compound? We tried this again with using multiple regressions. And we did it again with essential oils. Essential oils are very good natural products to work with because the composition can be known up to 99% many times with GCMS. That's the only natural compound we can have the full metabolomics of it. What about essential oils for Alzheimer and acetylcholinesterase inhibitory activity? Well, we've got a bunch of data from literature on the activity of these essential oils on this enzyme. And we apply multiple regressions. And we've got these 6 compounds. And amazingly, these 6 compounds, when we got them 1.5 years ago, all these 5 were known to have a direct inhibition of the enzyme. So let's say that from 200 compounds, the multiple regression already selected the candidates, the right candidates. Estragon was 1.5 years unknown, but half a year, 6 months before I'm talking now, it was reported by a group having the direct inhibitory activities on acetylcholinesterase. So that's a good tool also to explore. if you want to reduce the complexity and try to find clues and candidates for your complex natural product. Part 3 will show how we can do a translation in between Chinese and English using cluster analysis. Cluster analysis is another way to do multivariate statistics. Chinese have a very complex and very good traditional medicinal system. It works very well, and the proof is that the population is still growing and growing, and the first point of call is always the traditional medicine. But you know, our systems are completely different. This is how a medical doctor European sees a person, like a machine. You can, like a car, you can open it up and dismount the engine. It's like a machine. But the Chinese have a totally different approach. This is the Ayurveda, the Indians. You can see they are from the same year, about the year 1700. And we have different approaches. The Chinese will talk you about yin and yang, key energy, meridians. They have 5 elements. And you say, well, this is rubbish. This is just poetry. For the herbs, it's even complex because they have 4 properties: hot, warm, cool, they are maybe tasting, sour, bitter, sweet, and you have all the combinations there. And then again you say, well, this is just medieval. How can this work? Well, let's pay attention what we do in Europe. You know, what's light? And one say, it's a wave. Is it really like a wave? Is it like a wave in the ocean? Or is a particle? No, it's neither. There are metaphors. We also use metaphors. The Chinese talk about cold and hot and sour, and we talk about little balls or waves like in the ocean. We have to assign words to the model for it to work. We have to be above the words. So actually, we know that when they talk about earth, they will be talking about the spleen, or maybe blood will be liver. There's a correlation. My objective here was, because my expertise is in skin, cancer, and inflammation, is can we interpret the traditional Chinese medicine approach to skin inflammation the way we do it in Europe? In Europe, we believe that skin and lipoperoxidation is the link to diseases. Skin is damaged by irradiations, bacteria, or our pro-inflammatory cells, and this damages the membranes and produces an unspecific oxidation of phospholipids in the membranes, which is translated and computed by the cell, which responds with the selective oxidation. We have an unspecific oxidation and a specific oxidation as a response of arachidonic acid, and then they send a message of alarm. This is more or less what I'm talking about. This damaged the membrane with lipoperoxidation, and this produces malondialdehyde. This exerts into the DNA, which calls for the phospholipase A2 to liberate the arachidonic acid, produce several pro-inflammatory agents which go and call for help to the neutrophils, which in turn may be going to damage more the skin. And that's many times the vicious loop we have in dermatology, that you cannot get rid of any skin atopic eczema because this is a loop going on. There's also xanthine oxidase, which is a source of intracellular free radicals that do the same as malondialdehyde. Well, for 2 years I was in a project studying all the unspecific and specific oxidation inhibition of this set of randomly selected traditional Chinese medicinal plants. And during these 8 years, we've got a lot of data, which are represented here, and many more maybe, for inhibition of inhibition of xanthine oxidase, inhibition of lipoperoxidation, inhibition of malonyldehyde reactive products, and inhibition of each of these enzymes. If we put in a three-dimensional biochemical and pharmacological space all this data, we have here each plant and their activity in different enzymes of the arachidonate pathway, and here each plant and their activity in different lipidic oxidation processes. We have a distribution obviously in 3D, and 3D is difficult for the human eye sometimes. If we combine them, we have much more dimensions, and that's impossible to see. If we can reduce the dimensions by just plotting a module of the vector for each space, and then we have a single number for each set of 3. If we represent then the single numbers in a 2D space, eicosanoids inhibition here, antioxidant inhibition there, we have all this distribution of the medicinal plants the Chinese gave us. They are random. We don't know if they are good for skin or not. It was randomly designed. The human eye doesn't see doesn't make any sense of this, but the computer, if you ask for a cluster analysis, tells you this. And now it's easier it's clear that there were 3 groups. This group is inhibiting a lot both processes. This group is inhibiting a lot the eicosanoids, but not much the antioxidant lipoperoxidation, and this group is not very interesting as inhibiting anything. Let's put on top of it what the Chinese says about the temperature of the herbs. And suddenly we see that this group is only made of cold herbs. This group is made of hot or neutral. And this group that doesn't do anything, it's heterogeneous. Let's put on top another layer, and then these are the flavors. Actually, this group is not only cold, but they are all bitter. This group is not only warm or neutral, but also sweet. And these, they are all acting on the qi and the wind. So suddenly we have a way to translate what the Chinese are saying to the molecular biology we are in Europe proud of. If we go deeper and look at the clinical and pharmacological correlations of this, we have the cold and bitter group of very good inhibitory plants actually are used in multiple formula of traditional Chinese medicine for skin conditions. I remind you, I got a completely random set of plants. And they came out together, and these are the first point of call for the Chinese for skin. And they are called cleansing herbs, or yellow herbs, because they are all quite yellow, most of them. You'll see why. This group are warm and sweet, and but they don't have antioxidant inhibitors. And we have here the only fungi we have, and Angelica sinensis is separated from Angelica pubescens, so 2 plants from the same genus are separated actually, and then Astragalus membranaceus. Why this may be? Well, here, as I said, are called yellow because Coptis chinensis and Philodendron amurensis and Scutellaria bicalensis are yellow, not Forsythia suspensa. But these 2 are very far away in the botanical way to see at them, but they both have berberine alkaloids. So this approach also grouped up the chemistry in some way. This contains lanostains and Porreacoco too. They contain lanostain, even if this is a fungi and this is a plant. And why Angelica sinensis and pubescens are separated? Well, maybe because pubescens is the only Angelica producing furanocoumarins, which are not present in sinensis. So this bit of magic could be expanded, and we are working more on trying to input all the set of Chinese attributes and trying to reduce the multiple dimensions to extract more information from it. But the point is that actually we not only proved that our European concept of lipoperoxidation and skin works, because in a way could make sense for our knowledge and knowledge from Chinese, but also that by doing this, we can select plants from random sets and earmark them for future research or for using in future medicines. So I thank you very much again, the organizers, and you for your kind attention. Muito obrigado pela atenção. I'm ready for any questions you may have. I have to disclaim, I cannot predict the future.